How do you pronounce your name?
Willgate.
This is like almost 100 house. Yeah, so I just went and had some eating that on dad. Yeah. I mean, often this area and find a lot of water.
Right.
It starts to rot. Yeah, it's a good thing. Yeah.
This season, yeah, or this class, no, it came, you've covered other seasons.
No.
Yes.
But this is your question.
I have it here.
But I get the gist.
Oh, share my screen for.
Oh, she was.
Can you speak of the next hour?
Hello? Hello?
Sandy or Tom, can you tell me if the audio is working all right on this? Yeah.
Check, check, check.
And is your audio turned on or turned off?
My audio is turned on. And it's showing me speaking here.
Okay. So your audio is on, is your microphone on?
It should be. Hello?
Electric microphone, you need mic back. Go ahead, testing. So if you can speak a little bit.
Hello? Check, check, check.
Okay. Folks at home are saying it's good. We have next. Thank you very much for coming. Appreciate it.
1 second, Tom. It seems that my this won't move away from my screen.
You can click out of the trans. So it's not on your screen. I'm seeing your cursor up on the screen.
Yeah. Somebody dug all mine up there.
Oh, there we go. I can get rid of it here.
Well, I don't know. That's really done well. Yeah, it's really good.
Okay, we should be all good to go.
All right. You can stand right there. Oh, yeah, probably gets us all. Thanks everybody for coming today. I'm looking forward to this. I know about amoeba dysentery and amoebic brain infection. It's good to see a good side. Escort proteins and regulating plasma membranes. Frontiers of Life Sciences. What's the name of this talk? And then we'll be off on November 23rd because. And then on November 30th, we'll have our 10th and final presentation of the fall. And it's on the future of farm safety with John Schutsky from Biological Systems Engineering. So if you think it's cool to have vehicles around. Next week, Mark Kopelovich from the La Follette School in Political Science will talk about inflation, the global economy, and the consequences of the midterm elections. We'll be off on November 23rd. Also, November 30th, the sun is ready to make your electricity greener and cheaper in Wisconsin. That's Michael Arnold from Material Sciences and Engineering. And then on December 7th, Mitchell Ramuda from Cell and Molecular Biology will be talking about pulling viruses out of thin air. I used to pull viruses out of thick plants. It's a lot easier. All right. Welcome everybody at home to PLATO Frontiers in Life Sciences. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. We're here at UW Space Place for PLATO Frontiers in Life Sciences. Today, My pleasure to introduce to you Matt Wilgate. He's with the Department of Animal and Dairy Sciences. Matt, I'm going to ask you 5 questions. You get to answer them any way you'd like. Remember, they don't have to be true. They just have to be believable. Where were you born?
I was born in Oak Lawn, Illinois. It's a suburb right outside of Chicago.
Which direction?
It's southwest.
Southwest. Good. And then where'd you go to high school?
I went to Marist High School in Chicago.
And then where'd you go for your undergrad and what did you study?
I did my undergrad here at UW-Madison and studied biochemistry.
And now you're getting your PhD
Yes,
in what department?
in the Department of Animal and Dairy Sciences.
Very good. Today you're going to talk with us about harnessing soil-dwelling amoebas to combat incurable wound infections. Would you please join me in welcoming Matt to later?
Well, thanks, Tom, and everybody for having me here today. I'm not going to be talking about the brain-eating amoeba that Tom mentioned today, so I'm just warning you that. So today I will be talking about soil-dwelling amoeba as a potential therapeutic to combat bacterial biofilm infections. So as Tom mentioned, I'm a second-year PhD student at the University of Wisconsin-Madison in the Department of Animal and Dairy Sciences. I'm pursuing a doctorate in animal sciences, and my lab specializes in using swine models to study various diseases in humans. My research focuses on microbial infections, and today we're going to be talking about bacterial biofilms and amoeba. So show of hands, have any of you heard of bacterial biofilms or amoeba before? Okay, so a few of you. So this is a good start. So more specifically, as I mentioned, we're going to be talking about the therapeutic potential of using amoeba to combat incurable bacterial biofilm infections. And I'd like to begin today by acknowledging my lab and the bacteriology department at UW-Madison. I would also like to thank our industry collaborators for their contributions to this project. And I'd like to warn you all that the next slide is a bit graphic, and it's not for the faint of heart, but bacterial biofilm infections are a major global healthcare problem, and the severity of these infections needs to be recognized. These are untreatable bacterial biofilm infections. They're gruesome, painful, aggressive, and becoming rapidly fatal. This represents the problem that we're going to be talking about today, and most importantly, a problem that my research hopes to solve.
Matt, will your cursor close out that thing about who can see this transcript?
Yeah.
We have to look at it on your screen.
I don't have it on my screen. I'm trying to move the cursor over there, but can't.
You just have to slow it down.
Oh, wait a minute. There we go. Okay, so what's the culprit to these biofilm infections? Well, they're called bacteria, and I'm sure many of you are familiar with them. Bacteria are the oldest organisms and found in nearly every habitat in the world. Most of their DNA is contained in single chromosomes, but they also possess these small circular DNA molecules called plasmids. Plasmids replicate separately from the chromosome and can be shared across many different bacterial species. This primarily occurs by cell-to-cell contact. But plasmids are not essential for a bacterium's everyday survival. So why are they important? Why do bacteria use them? Well, the genes that plasmids carry often provide bacteria with numerous selective advantages, including antibiotic resistance, toxin production, and several other virulence and survival strategies. Most bacteria are harmless, but plasmids provide them with the opportunity to acquire new genes and become pathogenic. Consequently, this causes diseases in humans, plants, and animals. And we often think of bacterial cells as living in these free, single cellular states. And in reality, this is partially true. But most bacteria actually exist within these large microbial communities called biofilms. So what are biofilms? These are bacterial biofilms. The image on the top left represents a biofilm infection of the trachea. On the bottom right, a biofilm in a waterbed. And all the way on the right there, you can see biofilm colonizing the skin of a cow. As you can see, the bacterial cells are characterized by these spherical and spindle-like shapes, which I've highlighted in green, yellow, and pink. But there also seems to be something else here. There seems to be something holding these bacterial cells together, almost like a slime-like mesh. And this is a bacterial biofilm and the most important contributor to a bacterial biofilm infection. I'd like to pause for a moment and acknowledge that these are highly diverse communities of bacteria. And oftentimes, it's not just one bacterial species living in biofilms, but multiple, all contributing to each other's survival and infectious potential. Biofilms are the most predominant mode of life among all bacteria. In fact, 90% of bacteria exist as biofilms in nature. They form this protective barrier that separates bacteria from the dangers of their environment. You could almost think of them as castles or bunkers for bacterial cells. And although we are unable to see most biofilms with the naked eye, they're everywhere. They exist on our bodies, on our livestock, and even on some of the food that we consume. I don't know if any of you are kombucha drinkers here, but they're also abundantly present in those teas. As I mentioned before, most of what we learn about bacteria often focuses on them in these single-celled states, and also bacteria living in a free environment. But what about bacterial biofilms? What are they made up of? Well, those mesh-like structures between the bacterial cells that I pointed out are called extracellular polymeric substances, or the biofilm matrix, which are the primary components of a bacterial biofilm. Biofilms are commonly defined as an accumulation of microorganisms embedded within a self-producing extracellular matrix. This matrix is composed of polysaccharides, proteins, extracellular DNA, and lipids, which all contribute to the structural integrity of the biofilm and also the biofilm functions. So why do bacteria prefer producing and living within biofilms versus a planktonic state? One of the major reasons is that biofilms provide bacteria with this defensive armor, protecting them from antibiotics, harsh environmental conditions, predators, and several other environmental stressors. One of the major ways that biofilms offer these protective strategies is through diffusion inhibition, often carried out by enzymatic proteins and diverse polysaccharides within the biofilm. And this allows biofilms to almost become impenetrable. For example, antibiotics, in order for antibiotics to be effective against biofilms, you must increase their dose by nearly 1,000%. You would never give a patient anything close to this concentration of antibiotics because it would be extremely toxic to your cells and likely kill you. Biofilms also provide an environment where resistance genes are constantly swapped around, attributing to why they're one of the major causes of antimicrobial resistance. So let's say that you're a bacterium and you've just infected a wound of the skin. Antibiotics start pouring in, and the best opportunity for you to survive is to acquire a resistance gene from a neighboring bacterial cell. You have two options. You could continue living in a free planktonic state, such as the bacterial cells shown here, or you can form or join a biofilm. I can't walk all the way over there, but that's the biofilms all the way on the right side, where the bacteria are all embedded in that slime-like structure. So I'll give you a moment to think about your options here. And if you're thinking join or form a biofilm, then you're correct. I even gave you a small clue where you can see the antibiotic actually repelling off of the bacterial biofilm. And biofilms are often composed of several species of bacteria that cooperate with each other and help each other survive. A team of researchers were particularly interested in studying these cooperation strategies. And to do this, they placed 4 different bacteria strains on a corn leaf and observed their behavior. The results showed that each of the bacterial cells positively interacted with each other, acquiring new survival strategies that the bacteria themselves did not have. They also demonstrated that biofilm production was significantly elevated. Among each of these species, the bacteria produced nearly 300% more biofilm than they would have by themselves. So how do bacterial biofilms form? Well, the most common process can be broken down into the five stages illustrated here: motility, adhesion, maturation, dispersion, and propagation. And the process begins when free living bacterial cells adhere to a surface or other bacteria in their environment. To do this, bacteria utilize a wide range of adherence strategies. But it is only when bacterial biofilms begin to produce the biofilm extracellular matrix that the process becomes irreversible. And once the process becomes irreversible, the bacterial cells within the biofilm become hundreds to thousands of times more tolerant to antibiotics. So what's next? The bacterial cells are in a pretty comfortable position here. They're protected from predators in their environment, they're protected from antibiotics, and they're surrounded by other bacterial species that can prolong their survival. Why leave? Well, there are several reasons. Some biofilms may deplete the available nutrients in their environment, eroding over time, which causes the bacterial cells to seek out new locations. And from a survival perspective, this makes sense. It makes sense for bacteria to spread out and spread biofilms across their whole environment. This makes them become a more dominant species. And from a pathogenic standpoint, the bacteria within biofilms want to take advantage of their host. And by spreading their biofilms across different areas of the body, they're able to do so and take advantage of more host resources. Biofilms impact many different areas of our lives and are a major threat to a variety of global industries. These include food processing, agriculture, healthcare, waste treatment, and marine and aquatic engineering. The total economic burden that biofilms have on all of these industries approximates to $5 trillion per year. And although I don't have time today to go through the impact that biofilms have on each of these industries, I really want to emphasize that biofilms have a significant economic impact on our society, and it's crucial that we study them. The major industry that I would like to focus on today is healthcare. Bacterial biofilms pose a significant threat to the healthcare system. Among 35 to 40 million people that are admitted to the hospital each year, 5% of them will fall victim to a microbial infection. And among the 2 million infected, 100,000 of them will die. To give you an idea of the severity of this number, 40,000 people a year die from breast cancer. Hospital-acquired microbial infections more than double the number of breast cancer deaths. And among all of these diseases that cause these deaths, bacterial biofilm infections contribute to approximately 80% of them, with pneumonia and bloodstream infections being a leading cause. And I wish I could tell you that this number is declining, but it's not. One group of individuals that are particularly impacted by bacterial biofilms are diabetic patients. 1 in 4 people with diabetes will experience a diabetic foot ulcer. And these ulcers often lead to chronic infections due to factors that impair wound healing in diabetic patients. And the only way to treat these infections is often to remove a large piece of the tissue or amputate the foot. In fact, 10 Americans undergo an amputation every hour due to diabetes. And during the pandemic, this number rose by 50%. In a study that was just published this year, it was estimated that 4.9 million people in 2019 died due to antimicrobial resistant microbial infections. And 1.3 million of those deaths were a direct cause. This accounted for more deaths than AIDS and malaria. And it's now estimated that by 2050, 10 million people will die every year from drug-resistant microbial infections. And that number is going to surpass cancer, respiratory disease, and diabetes, which would make drug-resistant microbial infections a leading global cause of death. And one of the most challenging biofilm-associated infections are caused by a group of microorganisms known as Staphylococcus bacteria. Staphylococcus bacteria are common bacterial colonizers of the skin and mucous membranes in humans and animals. But their primary virulence factor that allow them to cause disease is biofilm formation, attributing to why they're the #1 cause of death from microbial infections acquired in the hospital. They're also the most frequently isolated organisms from hospital-acquired infections and a leading cause of catheter-related bloodstream infections and nosocomial sepsis. And again, I would like to warn everyone that the image on the next slide are graphic, but highlight the severity of these infections. Each of these photos represents a biofilm-associated infection, and Staphylococcus bacteria often produce biofilms in these susceptible areas of the body, such as wounds or surgical sites. And once biofilms are established within an infection, there are limited treatment options, often leading to sepsis or death. And although these infections are most commonly found in hospital settings, many of us are also at risk. These infections often emerge in our communities through physical contact, wounds, and injection drug use. And as antimicrobial resistance continues to emerge as a leading global health threat, the probability of acquiring one of these infections is only set to increase. So what else can we do to treat bacterial biofilm infections? When I first learned about biofilms, I became incredibly fascinated by them. I wanted to learn more about their development processes, their disease-causing abilities, and more specifically, how to eradicate them. And when I joined my lab, one of the ongoing projects focused on biofilms and identifying new therapeutic strategies to combat bacterial biofilm infections. Unlike most labs in the field, we weren't using these advanced chemical or pharmaceutical strategies to develop new therapeutics, but nature. As I mentioned before, bacteria are the oldest organisms on Earth. So why not go back in time and identify some of their first predators? Well, that's exactly what we did. And these organisms are called amoeba. Amoeba were one of the first bacterial predators and spend most of their lives killing and feeding on bacteria. I'd like to play a short clip for everyone showing amoeba actually feeding on bacterial cells. Amoeba are that larger structure there, and all those smaller spindle-like structures are the bacteria. And you can see that the amoeba chases down the bacterial cells and consumes them, even making a train of bacterial cells that it can consume later on.
Is this in real time or is it sped up?
It's a little bit sped up. So amoeba play major roles in regulating bacterial cell populations in our environment. But what if we can use amoeba in a different way? What if we can use them to target bacterial cells in an infection? Well, it turns out that I wasn't the only one interested in answering that question. Multiple UW-Madison professors have been involved in studying these organisms dating back to the 1950s. And because of this, UW-Madison is now home to a unique collection of 1,800 strains of amoeba known as dictyostelids. Dictyostelids are single-celled organisms that are commonly found in the soil. They spend most of their lives regulating bacterial populations in their environment. And to do this, they eat them. They make up one of the largest families of amoeba in the world. They're commonly referred to as cellular slime molds for their slime-like appearance, or social amoeba for their unique lifestyles. The most well-studied species of dictyostelids is known as Dictyostelium discoideum, and it was first discovered in a North Carolina forest in 1935 by a UW-Madison professor known as Kenneth Raper. The images here represent Dictyostelium discoideum in multiple different stages of its life. And here you could actually see them growing on tree bark. As I mentioned earlier, this species was identified from a forest, and you can commonly find these dictyostelids in the forest. And usually dictyostelids are observed as these macroscopic spores or cellular aggregates. And since their discovery, since the discovery of this specific species known as Dictyostelium discoideum, almost 150 more species have been identified across the world. Amoeba predominantly exist as unicellular eukaryotes. And typically, when we think of eukaryotes, we think of multicellular organisms, humans, plants, animals, and fungi. But dictyostelids possess the rare ability to transition between single cellular and multicellular states. The life cycle of dictyostelids is fascinating. Throughout their lives, free-living amoeba cells will primarily feed on bacteria. This is illustrated by the small amoeba cell in the top right there. But when amoeba run out of food in their environment, food meaning bacteria, they don't die, but rather they do something incredibly interesting. Each starving cell will produce a chemical signal, alerting other amoeba in their environment that their food supply has now been depleted. These signals induce a sudden transformation in the amoeba community, causing the cells to swarm together and form what is known as a mound or a slug. And although this slug appears as a multicellular organism, it's not. It actually represents a bunch of amoeba cells that cooperate together and mimic a multicellular organism to hunt down more bacteria in their environment. The amoeba form the slug as a survival strategy, allowing them to move around more efficiently than they would as single-celled amoeba. And if they're unable to find bacteria in their environment, the amoeba cells will go into a dormant state. But before this happens, the slug or mound will organize itself into a spore-forming fruiting body, which will protect the cells during their dormancy period. And once food becomes available again, these spores will germinate, releasing all of the amoeba cells back into their environment and restarting their life cycles. Now, I know this is quite the elaborate process, so I would like to play a short clip for you all to gain a better understanding of what's going on here. And before I start the video, I just want to point out that these small little dark specks here are all individual amoeba cells. And in this video, they just fed on all the bacteria around them. So right now they have no bacteria left. Watch what happens. So here they begin to swarm together. This represents that first stage of multicellularity. And now they're forming that mound that I mentioned. And now from that mound, something pretty incredible happens. They start to work together as slugs and travel throughout their environment, looking for more bacteria to feed on. And again, I would like to point out that these are single-celled organisms that just came together to mimic a multicellular organism to seek out more food for themselves as a survival strategy. It's a really fascinating process.
That's a 16-year-old video in 2006.
Yes, yes.
Good name that thing. It looks like a hurricane.
We could just stay on this slide for a while if you want to. Okay, but let's say that these slugs weren't able to find any bacteria in their environment. I mentioned to you that they start to form these spores. So on the next slide, I'm going to play a video of that spore formation process. That's that multicellular mound that they create. And here they begin to form these stalks that will suspend that those amoeba cells in that spore up into their environment. No, actually, the stalk is made up of amoeba cells. 20% of them will actually sacrifice their lives in order to support the other population to survive. They're actually one of the only organisms to do this. But what's the reason behind creating this stalk and suspending these cells up into their environment? Well, as I mentioned, amoeba live in terrestrial environments. So they're surrounded by many animals and insects. So these spores will actually latch on to animals or insects and take those cells to a different environment where bacteria are more abundant. And obviously further than the slug could go. So that's why they do this. So all the videos that I've showed you so far have involved amoeba feeding on free-living single bacterial cells. But as I mentioned earlier, 90% of bacteria exist as biofilms. So how have amoeba dealt with this? Have they evolved mechanisms to penetrate biofilms and feed on the bacterial cells inside? Well, one of our collaborators and former UW professor, Marcin Falutowicz, who unfortunately passed away this year, was one of the first individuals to show that dictyostelids penetrate biofilms and feed on the biofilm-embedded bacteria. The videos here are from his 2017 publication demonstrating this process, and the large masses here represent biofilms, and the little dark specks are amoeba cells. As you can see, they all start to attack the biofilm, but are they able to penetrate it? Well, as you can see, the biofilm is getting much lighter, which means it's actually being removed. It actually turns out that they eradicate the entire biofilm. And at the end of this, you can actually see them forming that swarm again. Pretty incredible, huh? Now, I would like to remind everybody here that there are very limited treatment options against bacterial biofilms, not only in hospital and healthcare settings, but also in the environment. Yet these small little dictyostelid amoeba cells just seem to eradicate them effortlessly. Now, as an incoming graduate student, you could only imagine how excited I was to have access to UW-Madison's 1,800 strains of these amoeba, most of which have never been studied before. Is it possible that they've all acquired mechanisms to destroy biofilms? And how do different dictyostelid or bacterial species influence this process? Well, here's some of the first data that we developed. And these images represent untreated Staphylococcus biofilms and biofilms treated with dictyostelid amoeba cells. The extracellular matrix is difficult to see here, but it's represented in these very dark structures between the cells, and it mimics what I showed you on one of those first slides. That represents the extracellular matrix of the biofilm. So what question were we asking here? Well, we know that dictyostelids could feed on the common bacteria that they encounter in their environment, but what about bacterial pathogens? Our observations here were actually astonishing to us. The dictyostelid cells, indicated by the white arrows, not only seem to destroy the biofilm matrix, but also eradicate the bacterial cells inside. Now, I'd like to pause for a moment and have you all think about this. This is a clinical isolate of Staphylococcus bacteria that these amoeba cells have never seen before in their lives. In fact, this amoeba strain that we tested this on was isolated in 1985 and has since gone extinct. We're actually the only lab to possess this amoeba strain that is able to feed on this biofilm. And yet, these amoeba were still capable of destroying that bacteria, a bacteria that they'd otherwise probably never encounter in their lives. From a therapeutic perspective, we knew that there might be drawbacks to using a live organism to treat bacterial biofilm infections. And although we're still looking at this and seeing if amoeba can actually destroy biofilms in a wound, we decided to ask a different question here. Are the amoeba producing compounds or inducing the production of compounds that are capable of degrading biofilms? And to set up this experiment, we established these Staphylococcus biofilms on porous membranes, and then we treated them with dictyostellate amoeba cells. We waited for them to feed on all the bacteria, and then they produced these spores. And from this, we then collected the secretory compounds that were produced during that feeding process. And once we separated the spores from the residual bacterial cells, once we separated the spores and the residual bacterial cells from the secretion extract, which I will now refer to as dictyostellate-induced antibiofilm compounds, or DIABC. Just know when I say DIABC, I'm talking about these compounds that we isolated. And then we applied them to freshly grown Staphylococcus biofilms and tested for antibiofilm activity. So these two images here represent fully established Staphylococcus biofilms on these membranes. And the two images on the right represent the biofilms that we treated with these compounds. As you can see, the biofilms were completely eradicated. And again, at this point, the amoeba cells are gone. We're only using some of these secretory products that they released when they fed on the bacteria. We also performed another antibiofilm assay to confirm our results, and both supported each other in finding that these compounds are actually able to degrade biofilms. We were also interested in assessing the antibiofilm activity at different time points, and we found that these compounds were capable of destroying biofilms in less than 2 hours.
When you talk about destroying biofilms, are you also talking about destroying the bacteria? Or
So
is the bacteria left after the biofilms?
yes, so right now we're actually looking at, so usually when dictyostellates feed on bacteria, they kill them. So currently we're looking at whether these compounds are actually capable of killing the bacterial cells or just destroying the biofilms and releasing them into the environment. Because you would think that the dictyostellates actually just release compounds to open up that biofilm so that they can feed on the bacteria. So to further assess the reduction of Staphylococcus biofilms, we generated these scanning electron microscopy images. And one of the major differences between these two images is the presence of the extracellular polymeric substances, the primary components of bacterial biofilms that we discussed earlier. The image for the untreated biofilm control group is highly consistent with a biofilm phenotype, but the image of our treated compounds is not, suggesting that the amoeba are targeting the extracellular matrix components of the biofilms to release the bacterial cells so that they can feed on them. And I would also like to note that without the protection of this biofilm matrix, the bacterial cells are no longer protected. And what this means is that bacterial cells now become susceptible to antibiotics and also our immune systems. One of the problems with biofilm infections is that our immune systems can't recognize that there's an infection there because the matrix actually has this evasion mechanism where our immune cells can't detect that infection. So what I'm saying is that these compounds are actually capable of destroying the matrix that protects the cells. And there's just huge therapeutic potential here. We're also interested in investigating what kind of compounds were causing antibiofilm activity in the extract that we isolated. And to do this, we fractionated the compounds down by size, separating the small molecules from the proteins, and then applying them to Staphylococcus biofilms. So I'd like to walk through this a little bit. So here are Staphylococcus biofilms that are not treated, and the image next to it are the original compounds that we isolated. And what we did was we took these compounds isolated them down further, separating the large molecules in the solution from the small molecules. And the idea for doing this was that we wanted to find out whether, is this a small molecule acting upon these biofilms or are there proteins? And interestingly, when we separated those compounds, the small molecule fraction completely lost that anti-biofilm activity, but the protein fraction didn't. So what this suggested is that amoeba are either inducing the production of or producing these compounds themselves. And they turn out to be proteins or macromolecules. And furthermore, we performed mass spectrometry, which is able to actually identify proteins present in a solution. And we identified the potential proteins that amoeba actually target in bacteria to disrupt biofilm formation. And although we have not identified the specific protein or proteins responsible for anti-biofilm activity, this work is still ongoing. So what's next? As I mentioned before, it's often not one species of bacteria living inside of these biofilms, but multiple. And although we've shown that these compounds are effective against pathogenic monospecies biofilms, many of these severe infections contain several different species of bacteria. So is it possible for amoeba to also penetrate these kinds of biofilms? Well, I have some preliminary data to suggest so. So here represents two species of Staphylococcus bacteria that cause infection. Right here you have Staphylococcus epidermidis, which I grew the biofilm separately here. And then I grew Staphylococcus aureus, again, separately. But what a polymicrobial biofilm is a biofilm full of different species of bacteria. And you can imagine that these bacteria are common colonizers of our skin, and they're also found in infections. So what I did was combine Staphylococcus epidermidis with Staphylococcus aureus, and then tested our compounds again to see if these compounds are still able to degrade now a multi-species biofilm. And based on this data, it looks like they can. And remember that these compounds that we've discussed so far are only isolated from a single dictyosteliid and Staphylococcus bacterial species. So what happens if more species of dictyostelids and bacteria can produce these compounds? What happens if I can isolate more of these compounds with this large collection that UW-Madison has of these amoeba? Well, again, I have some preliminary data that suggests that multiple strains of dictyostelids can produce these anti-biofilm compounds. And you don't have to understand the entirety of this data set, but let me just summarize it a little bit for you. So it appears that different amoeba species have a preference for their bacterial prey. And they also have a preference for the secretions that the compounds that they release to destroy biofilms. And it doesn't matter whether these compounds are produced by one species or multiple species of dictyostelids to degrade biofilms. And this suggests that we might be able to actually produce a super cocktail of these dictyostelid anti-biofilm compounds that can destroy multiple different species of these bacterial biofilms. In addition to investigating these different combinations of these compounds, we're also now looking at the efficacy and safety of using these compounds in actual wound environments. And we hope to develop a new therapeutic in the near future. So we believe that these dictyosteliid amoeba offer a new and exciting approach to combating incurable chronic bacterial biofilm infections that have never been, and these processes have never been seen before. In addition to producing or inducing the production of these anti-biofilm compounds, amoeba may offer new mechanistic approaches. There are currently no known therapeutic target sites on bacterial biofilms, and understanding the mechanisms of how dictyostelids degrade biofilms could be a groundbreaking discovery, similar to the discovery of antibiotics. Thank you all for your time today.
Question?
Yeah.
Yep, the back first.
how do you keep alive the amoeba that you have in the lab?
So typically when we want to perform experiments, we'll take the amoeba out from a stock that we have. So we have isolated several spores of these different amoeba species, and these spores are very stable. So you can actually keep these spores for a long time. The strain that I showed you was a strain from 1985, and we still have it in the lab. So what we do is make a bunch of stocks of these amoeba spores. And when we have an experiment, all we have to do is apply them to bacteria. And usually we use common bacteria that they're familiar with in their environment. But that's one of the differences here, is that I'm actually starting to use pathogenic strains and seeing that they can feed on them similarly.
Yeah, you mentioned how the biofilms can kind of make a defense against antibiotics and immune chemicals. So how about bacteriophages, viruses? Does the biofilms
So-
keep the viruses out too, or?
Yeah, so there's a lot of new research going on with bacteriophages as a new therapeutic to target biofilms. And a lot of the work is really promising, but some bacteriophages are actually shown to penetrate biofilms. And there's a lot of potential therapeutic strategies being developed right now with those bacteriophages. And although I haven't worked with bacteriophages before, I think it would be really interesting to look at that interaction with these amoeba.
Any other questions?
So I noticed another thing I see is like wounds and stuff being cleaned up, they put the maggots in there. Is that just to clean out the necrotic tissue, or do those maggots actually do something with the biofilm, or do they know the bacteria is in there to get through the biofilm?
I'm not too familiar with that therapeutic strategy. I don't think it's commonly used anymore. But yeah, I'm not too familiar with the that mechanism there. So
I just see little videos and stuff every once in a while. These nasty wounds getting cleaned up, and that's just wondering, the maggots.
yeah, I would say, I think they're actually leeches, but they're I think they're one of the only live organisms actually used for therapeutic applications.
Is the biofilm the main or the only reason that bacteria are resistant?
No, there are several bacteria species that are just antibiotic resistant as single cells, but biofilms contribute to this antimicrobial process. So the faster that we can treat these biofilm infections, the more that we're going to limit the antimicrobial resistance that some of those bacterial cells actually in the biofilm don't have yet. So that's why when these infections are established, they become so dangerous, because you can't eradicate the biofilm, and now all the bacteria within are resistant to antibiotics even when they're not in a biofilm. So it's definitely a challenging topic.
Do benign bacteria make these biofilms also, or is it just seem to be the harmful ones?
Benign bacteria also make these biofilms in their environments. So these are a protection strategy for a lot of bacteria in the outside world, but also they now use it as a primary virulence factor to cause disease. So there's actually many different beneficial applications to biofilms too. So it's not only that biofilms are bad, but for my research, they're bad. So.
Are the spores like a next generation, or are they the same amoeba?
So they will be the same amoeba, and I didn't go into the other life cycle. So amoeba actually exhibit 3 different life cycles. There's actually a sexual phase where they actually reproduce. And I didn't think that was relevant to today's talk, but there's a whole different process for how amoeba sexually reproduce. But they are eukaryotic organisms and do sexually reproduce.
What you're showing us there are haploid?
I'm sorry?
The cells you're showing us are primarily haploid?
Yes.
Do the diploids do anything besides go through meiosis right now?
Not to my knowledge.
You had 3 or 4 patents listed on one slide. Did I read that correctly?
Yes, so that professor who originally found that the amoeba are able to penetrate biofilms ended up starting a company for these amoeba cells and looking at different applications that can be used, such as like potato rot. A common problem in agriculture is potato rot, where you get these diseases on potatoes. And one of the primary projects that he was looking into is using these amoeba to combat those, more of an agricultural application of using these amoeba. But he is the one who developed these patents, and my professor and a few others in my lab have been involved in.
And was that Raper or was that somebody else?
That was Martian Fludowitz.
For the treatment of the potato rot, are the amoebas applied topically?
Yes, so he was primarily interested in using the live organisms. And shortly before he died, he gained more of an interest when we showed him that these amoeba actually produce compounds. So instead of applying a bunch of live amoeba to crops, why not try and isolate these compounds first? I don't know if a lot of people would want to eat potato with a bunch of amoeba on them, but
Jerry would.
Once the rot's gone, what they do?
yeah. But another thing is that there are some amoeba species that cause disease, but these group of dictyostelids none of them have been shown to be pathogenic. So that's why we're also investigating the therapeutic potential of using the live organism, because they've never been shown to cause disease. And some of the research that I'm doing now is actually introducing different immune cells to them and seeing if there are any toxic effects.
Would it be quicker, and I realize this whole thing is a long term, but would it be quicker to use the amoebas themselves as far as getting it to a therapeutic point as opposed to finding the compounds and then figuring that out?
I think the regulatory processes that would be involved with getting a live organism to be used in a clinic is very complicated nowadays. Again, like I said, I think the leech is the only other organism that I could think of that has been used therapeutically, and it's not commonly used anymore. So even though it's going to be, even though there's a lot more work to do in identifying that protein, I think we're better off identifying that protein, because then we can maybe make that protein ourselves and not have to use the amoeba at all.
Well, I would think that would be the point of commercializing it.
Exactly.
Matt, would you like to introduce the folks that are here from your lab?
Yes, so this is my primary advisor, Dr. Danu, and this is a senior scientist in my lab, Dr. Taeyoung Shin. That is Jen Might over there. She is a former research manager of my lab, and that is our other PhD candidate in our lab, Nathan Chesmore, who originally trained me to start this work. Nathan is finishing up his PhD and was actually a large part of a majority of this work.
So did they get to evaluate the talk?
Yeah, I'm gonna run out of here when you guys get.
Thanks for coming to the talk.
Very interesting.
Oh, yeah, thank you for all your time. I really enjoy talking about my research, so I'm glad that you all are happy with it.
It shows. Yes.